plants. We are here. We are going to focus on adaptations of birds, but yeah, we have several examples. For example, morphological adaptation of birds that birds tend to adapt to be less. To fly less, for example, and shorten their wings or longer their tarsus. Or, for example, other adaptations in behavior, species tend to be less aggressive, or other kind of traits like signaling, also less colorful species on islands. So here, one of the most pattern of adaptation of animals in birds, one of the most known is the island rule that was described in mammals, where species that are big in the mainland when colonize an island tend to decrease in size, probably because of the resource limitation, while species that are small in the mainland when colonize an island became bigger because of the release from predators and competitor pressure. So here we wanted to test this pattern, but using birds. So we are going to follow this kind of graph here, where we compare the body size of the mainland species with the differences between body size of the island species and the mainland sister species. So if a species in the mainland it's bigger than a species on island is bigger than the mainland, we will find that this index is higher to 0, so it's a giantism. And if this index is below to 0, it means that the species had decreased in size in the island. So to find the island rule, we need to find this kind of pattern, this negative pattern, where species that are small became larger and species that are big become smaller. So we expect here that this pattern increase or is more strong when there are not predators or competitors on the islands. So this is what we expect, because when we expect that island that has predators or competitors are more mainland-like, so these evolutionary patterns of body size will be less evident. So to test this, we used 120 pairs of island and mainland bird species that are sister species. And you can see here that we sample species from different archipelagos and islands through the world. We only used endemic species from island, and then we compared with the sister species on the mainland. And we used the body mass information for both females and males individuals that we then averaged them for comparing from species. To take into account the predator pressure, we counted the number of raptors present on the islands and that predate on birds, because there are some raptors that don't predate birds, so we didn't account for them. And also we account for the prey-predator body masses relationship, because of course, probably there are some species that are big enough that won't predate on very small birds, or other way around. Regarding the competitor pressure, we counted the number of species of the same genus or the same family that are present in the island, because we expected that closely related species would share similar ecological niche and also habitat or trophic feeding, so may compete between each other. So then we do the analysis. We basically first calculated the island rule index here, as I said, by dividing the mass of the island species divided by the mass of the mainland species. And we calculated these models. The first simple model was basically to see if there's a trend of island rule on birds. And the others were to see if there's an effect of presence or absence of predators and competitors. And then we control all the models with the latitude, because there's no that the body size or body mass could be influenced by the latitude because of the Bergman rule. So what we find is that actually there's island rule happens in birds. So small species here become larger on islands, while big species become smaller on islands. But when we include predators, we find that when the predators are absent, well, there's still island rule, but when the predators are present, there's no longer this relationship. So probably it means that when the predators are present, well, this pressure prevents species to evolve to different body sizes. But when we explore the competitors, when we use the competitors as species that belong to the same genus, we don't find any different in the trend. So we found very similar trends. But when we use the competitors as species from the same family, we see that the species that occur in island, that there are no species belonging to the same family, so no competitors, this relationship is stronger, while when there are competitors, is less strong. So probably here there's an effect of competitors too, but we need to define better what a competitor is in the future, but it's quite difficult sometimes because we don't have enough information for the ecology of each bird on island, especially of endemic birds, but it's something that we want to explore in the future. So concluding, we can say here that birds follow the island rule like mammals, that the island rule happens only when no predators are present on islands. No differences in the island rule was found when considering co-occurring species belonging to the same genus. And the island rule is stronger when no species belonging to the same family co-occur in the same island. So, as I said, maybe we need to explore what is better, what a competitor is, and explore in the future if there's another way to define this competitor and see if there's an effect on the body size evolution of the birds on islands. So I want to thank you all for organizing this Congress. And if you want to see other projects that we have ongoing, you can visit this webpage, Island Bird Project, or contact me directly. And thanks a lot.
Excellent, thank you. Questions? Dick Titan, do you have a question?
Hello.
Hello? Dick, can you unmute if you have a question? Oh, you're still muted. Oh. Oh.
Oh, no question.
There's no question.
Okay.
Anyone else? I have a question. So
Okay.
if you see this, the island rule of changing body size where small birds get bigger and big birds get smaller, that also implies that for that ecological context, maybe on the island, there's a single body size that's favored that they get released to when they get onto the island. Have you looked at what just the absolute body sizes without correcting for what their sister size is and for any sort of phylogenetic correction to see if the variance of bird sizes on islands gets much smaller than on continental areas?
Well, we did when we built the models, we corrected the models with the phylogeny, of course. So maybe in that case, we can suppose that they are corrected by this kind of variance. But yeah, we didn't do any other extra correction regarding. Well, we compared the sister species of the mainland and the island, so by calculating this kind of index. But yeah, we didn't do any other extra correction, just considering the phylogeny in the models.
Well, I was just asking whether if you just look at raw body sizes, because the ecology of the system could really care less about what the phylogeny of all the species are.
Yeah.
If you just look at body size, do all the birds across the world on islands that show this relationship converge to a very similar body size? That implies there's an adaptive peak in that ecological context that all these birds have evolved to. And that's what's driving the pattern.
Yeah, this is something that we need to look into because it's super interesting. And yes, there's a tendency of a mean size on island, so a medium size, you know. And yes, it's something that maybe we should include in the future. Yes, thank you.
Other questions? Still got a couple minutes. Marco, why don't you end your screen sharing so we can get the next talk queued up?
Okay. Thanks.
There we go. Any more questions for Raquel? We've still got a couple of minutes, so let's wait for a minute.
I think there's one more question from Melanie.
Oh, there's a question. Yes, Melanie, go ahead.
Hi, so can you hear me?
Yes.
Yes.
Yeah, hi, great talk. I was just curious, so you assumed that competitors were species that were closely related, right?
Right.
Okay, so do you have an idea or can you give a sort of an explanation as to why species that were potentially more distantly related, so possibly less closely related, were, you know, showing a different trend to the species that were more closely related?
Yeah, well, this is something that we were discussing a lot before doing this work, because it's super complicated to see what a competitor is, or what consider to which you know, which what consider to see if a species is a competitor. So what we assume is that when species that are closely correlated, they probably would have similar ecological niche, also to similar trophic niche. But we cannot assume that species that are not closely related have similar niches as well, because there are, for example, species that are both insectivorous and they are not closely related. But this is something that we decided to do and to assume, to have a proxy of competitors, because we don't have enough information for all the species that inhabit in an island to say if there's actually a competitor or not. If we do this only in an island with some particular species, maybe we could, but in a global scale, it's super difficult. So this is why there's something that we need to explore in the future. Yeah.
Oh, thanks.
Thank you.
Thank
And
you.
our next talk is by Daniel Schoenberger. Daniel, take it away.
Yeah, hey, can you hear me?
Yes.
Perfect.
Very good.
So yeah, welcome to my talk. I hope I can give you a short introduction into the macroevolution of cell, nucleus, and genome size today. And first of all, we start thinking about an animal such as this beautiful dragonfly, one of my favorite insects. I think we can all agree upon that genome size, cell size, and nucleus size are fundamental characters that make out the size, the shape, and the fitness of this organism. However, then it's not really clear how these three traits are related to each other. What are the mechanistic links behind that? For many, many years, it has been assumed, and I think it's still the assumption that these three traits are basically all positively correlated, strongly correlated with each other. But again, it's not really clear what's the mechanistic link behind that. For example, I think it's quite logical that we could assume that if you have a genome size, a specific amount of DNA in a cell, this determines the nucleus size, because if you have a lot of DNA, you need a bigger nucleus. And then if you have a bigger nucleus, this basically determines also a bigger cell size. So that could be one way to think about it. However, there aren't really any studies that test this. And there's one hypothesis, so-called limiting pool hypothesis, from this study here. They used yeast experiments, and they found actually that cell size determines nucleus size, and that's basically based on the nuclear volume is determined by the amount of resources that are around in the cell cytoplasm, so around the nucleus basically. That's their hypothesis, and they also showed or proposed that actually genome size determines cell size, and that's basically based on if you think of cell divisions and mitosis, the size of the subsequent daughter cells is determined by the amount of DNA and the ploidy level in the cell before that, basically. Then there's a different hypothesis, the so-called nucleoskeletal theory, which is quite prevalent. And this basically focuses on genome size and cell and nucleus size. And as I said before, that basically is based on if you have a lot of DNA in the cell, and if it's not compacted at all, this means we need a bigger nucleus for that. A related principle is this so-called karyoplasmic ratio. It's basically if we plot the nucleus to cell ratio on cell volume, this is assumed to be constant. This basically means that if you have a small cell, you have a small nucleus, and if you have a large cell, you have a proportionally also larger nucleus. However, then there's this really cool recent study from 2021, and they looked across many species, from prokaryotes to mammals, and they found actually this is not constant, but it's negative, meaning that larger cells actually have relatively smaller nuclei. And the cool thing is that they also found that within species, so looking at several different species and what's the pattern within of these species, and they found basically the same pattern. Lastly, they also looked into an artificial selection experiment. So they selected different cells of this alga here for 450 generations for different sizes. And again, they find basically the same trend across these cells, that larger cells have relatively smaller nuclei. So that's one of the larger papers or larger research studies that looked into this pattern. However, it's still not really clear what's going on. They also show that actually cell size variation is much larger, as you can see here from these numbers, than nucleus size variation. And this basically indicates that cell size seems to be less constrained than nucleus size. Or it could also indicate that there's stronger stabilizing selection going on nucleus size compared to cell size. And then one last thing they also looked into is the correlation between nucleus and genome size. And again, they show across many species that actually this is quite strong positive correlation as we would expect. But I guess it's still cool to see that this pattern actually holds across many different species. And now me and my colleagues at Lund University, we were interested in this question using dragonflies and damselflies. So these really cool and bright insects here. And we were interested to see how are these traits related to each other, and what is the macroevolution of these traits, so over long evolutionary timescales. And one hypothesis I had was that if these three traits are all correlated with each other, they have strong correlations as we all assume. That basically means that we can also assume that they evolve in a coordinated fashion, right? Because if there's a shift in genome size, we would expect that cell and nucleus size show also a shift on the tree. And then we were also interested to see, is the evolution gradual, or are there any rapid shifts going on? And for that, we used a different project, basically, that was also using this data here. So we estimated cell and nucleus size based on this high-resolution imaging system of blood cells, hemolymph cells of dragonflies. And that's basically done with this operator machine here, which can estimate these cell and nucleus sizes for thousands of cells within an individual. So that's really cool. And here you can see some of these cells that really look like little galaxies when they're in this wet dye. So really, really cool. And then we used phylogenetic comparative methods based on these really nice books here that I can recommend. And first of all, we looked into the relationship between these three traits on the y-axis and body mass. And we can see, for example, cell size here, it's moderately correlated. But then if we go to nucleus size and genome size, the correlation is getting smaller and smaller. And actually, if we use phylogenetic path analysis, so that's basically a tool to indirectly infer mechanistic links between traits without doing experiments. And so that's again from a different project, but we can see that actually if we look at body mass, there's no link between body mass and cell size, or no link between body mass and nucleus size. So no arrow going this way. And that basically means that nucleus size and cell size don't seem to be affected from body mass or the other way around. And that's, I think, quite surprising, because if you think of a small and a large dragonfly, maybe you would expect that a larger dragonfly has larger cells. But instead, it seems to be that this larger dragonfly is just built of many, many more cells that all have the same size. And we can also see that nucleus size and cell size are strongly correlated. So it's not only that cell size determines nucleus size, but it seems to be basically both ways. And then we looked into the macroevolution using maximum likelihood models, using the AUI and PhyTools package, and also using reversible jump MCMC models in Bayou. And first we looked into genome size. So here we used published data and analyzed this. And we can see, so in light colors, we see really low genome size, and in darker colors, higher genome size. So there's large variation within the tree that is going from around 237 million years ago to today. And the interesting point is that we actually found evidence of long-term constraints and really strong pullback force, meaning that if you have large or really small values of genome size that are constrained at the upper or lower limit. And then we were also interested to see if flyers or perchers have different genome sizes. So flyers are, as the name says, they fly a lot, so they're mostly on their wings, whereas perchers, they're mostly sit and wait predators. And we are interested to see, have these flyer evolved smaller genome sizes? And this hypothesis is basically based on the observation for many other studies that show that flying birds and bats actually have smaller genome sizes compared to their flightless relatives. And they proposed that this pattern actually is based on that these flying birds and bats, they actually have evolved smaller genomes due to their higher activity and higher metabolic rates. And we wanted to test this on dragonflies and damselflies. So we actually found evidence for this multi-regime OU model. Basically, that means that both behaviors, they seem to be evolving towards different genome sizes consistently across the tree. And the surprising thing is that we actually found that it seems to be completely the opposite way. So actually flyers evolve towards larger genome sizes than perchers. So that's completely the opposite than we would expect based on observations from mammals and birds. And this might be related to the fact that we see that genome size seems to be genome size, nucleus size, and cell size is probably not too much linked to body mass in these insects. And I think in mammals and birds, it's the case that it's quite linked. And another study that we are hopefully soon publishing is that the metabolism and the metabolic allometry in these insects is completely different, so that this link between metabolic rate and genome size is also probably not there, leading to this completely different pattern here. And we also found multiple adaptive shifts here. Each of these dots is an adaptive shift, meaning that this clade where this adaptive shift is happening, they evolve towards different, either higher or smaller genome size compared to their background weight. So there are multiple different adaptive optima in genome size. Then we looked at cell size. So here on the left, cell size across only 20 species, but 7 different families. And actually here we can see that the evolution is gradual based on a Brownian motion model. So here there's no really evidence of long-term stabilizing selection. And we found the same thing for nucleosides, so gradual evolution. But here we found that actually behavior again seems to have a strong impact on this factor, meaning that actually perchers have evolved larger and they evolve faster nucleus. They have a faster evolution of nucleosides basically compared to flyers. Yeah, so again, there was basically no evidence of long-term stabilizing selection and evidence of gradual evolution in these 2 traits, cell size and nucleosize.
3 minutes.
Yeah. And that basically means that if we compare these 3 traits and their macroevolution, we have seen that genome size actually has evidence of constraining forces going on and also a strong link to behavior and multiple shifts, where cell size and nucleosize, they're closely related in terms of how they evolve, but it seems like there's no evidence of constraints and no shifts going on. So there seems to be that these 3 traits don't really evolve in a coordinated fashion as we assume, but something different seems to be going on. And again, there was a strong link to behavior in these insects. So there seems to be a difference between flyers and perchers, which is quite interesting to see. And with that, I want to thank all my collaborators, mostly from Lund University, and I'm happy to take any questions now.
Excellent. Questions? Janina has a question.
Hi, Daniel. Thank you so much for your talk.
Yeah.
So I have a question related to genome size and your observed patterns. And I would like to know, did you also incorporate, for example, transposable elements or repeats in general? Because it's known in many species that transposable elements are definitely positively correlated to genome size. So this could also be underlying what you see, but I'm not sure how this actually relates to nucleosides or cell size in general.
Yeah, I think like with one of my collaborators, we want to include these. And the only problem is that for these dragonflies and damselflies, like whole genomes are not really available for many species. So we think we can only use like 10 or 20 species to analyze transposable elements and maybe more genomic characters basically to look into that. And the problem is also that we can't really link cell and nucleosize to genome size because they are 2 different data sets, unfortunately. But yeah, it's definitely one thing to look into that. They seem to have a large influence, definitely.
Yeah.
Other questions? Oh, here's a question in the chat. The long branches separating dragonflies and damselflies would suggest a good bit of extinction. It's a really old group too. Do you have any sense of whether and how this could affect your results?
I mean, I wouldn't expect that this would kind of affect the results in some way. I think it's mostly if you look into how this affects maybe diversification rates or something, that then we would expect this fall into that.
If you redid the analyses with anisopterans and zygopterans as independent analyses, do you get the same answer?
Yeah, we get basically the same answer. Also the thing is that if you look into the 2 behaviors that we have, damselflies are mostly perchers, especially in our analysis here. So if we exclude these, we still find the same results basically. Yeah.
Excellent. So that is the final talk in this session. If you're in North America, go eat lunch. If you're not in North America, do whatever you do at this time of day, and we'll see you in the next section.